I'm really excited to be here to get to represent my whole team back at NASA Johnson Space Center and talk about how we're really pushing forward nanopore sequencing in spaceflight. So NASA's been relying on culture-based methods to do microbial monitoring in spaceflight for over 60 years. This is what we used with Apollo, and it's what we're still using on the ISS today. And a big concern that we have with this is it requires samples to be returned to the ground to be able to complete NASA's microbial risk assessments. And as we go towards deep space missions, like returning to the moon, we can't rely on samples being quickly returned to the ground to do those assessments. So that's why my team has been using nanopore sequencing in spaceflight since 2016 and developing these methods. And I'm excited to let everyone know that we will be launching the MinION Mark 1C as part of the Gateway Artemis program to do our future microbial monitoring. But what I'm going to talk about today is further about the monitoring that we do on the ISS and how we've implemented nanopore sequencing. So briefly, the crew does quarterly monitoring of air surfaces and water. And they culture them onto auger plates, and they grow them up on orbit, and they can look at them and say, there's a lot there, there's a little bit there, but we don't know what it is until it gets returned to the ground. And so it comes to our laboratory at NASA Johnson Space Center, and the isolates get identified using Sanger sequencing and Vitek biochemical assays. And this is what we've been doing since 2000. And so we have over 22 years of data showing that there are 32 bacterial genera with 87 distinct species, the top ones being Staphylococcus, Cronibacterium, Bacillus, and Micrococcus. And for fungus, we've seen 22 fungal genera with 16 distinct species, the top ones being Penicillium, Aspergillus, and Rhodotoila. And so this has us immediately wondering, are our microbial controls just working really well in the environment, or is our assessment of the environment just really limited by our culture-based methods? So this is where we've implemented nanopore sequencing to go to a culture-independent approach. So I'm really fortunate to be a core member of the team that's been doing sequencing in spaceflight since 2016. We've had a lot of really amazing opportunities to move this forward. We've supported 11 launches to the space station. We actually have a 12th one coming up soon this summer. We've turned 9 astronauts into molecular biologists and done 29 fundamental paradigm-shifting spaceflight sequencing experiments. This has gone far and beyond just the first DNA or RNA sequencing. We have full sample preparation methods that we validated in spaceflight. We've identified bacteria that's been fully collected and cultured off of Earth, and we have this full culture-independent workflow that I'm going to talk about more today. So we call that swab-to-sequencer, and that's what we're using for our in situ microbial monitoring. And briefly, basically the crew collects a swab from a surface, and instead of putting that onto a culture plate, they extract the DNA directly from the swab using heat and enzymes. They can clean it up with magnetic beads, and then we do the rapid 16S barcoding kit and clean it up and sequence it all on board. And we've spent many years developing and validating this, and it started out as a research experiment, and NASA saw that it was successful and saw the value in it. So they asked us, could we make this an operational activity, which is a really big deal to say, can the crew use nanopore sequencing operationally instead of the culture-based methods that we've been using for over 22 years? So we had this study called Biomole, and we got to work with 4 great crew members. We had Rajachari, Chris Cassidy, Aki Hashida, and Dr. Kate Rubens on her second mission, so we got to work with her twice. Here's some brief results from that study. So it was a culture to nanopore direct comparison. And so the slide in the top left corner is the contact slide of the plate that we got back from the space station with the cultured organisms. And then the picture shows where the crew member swabbed approximately 100 squared centimeters, and then the colored dots represent the nanopore IDs. Overall, we saw similarities between the IDs that we got from the culture data to what IDs we saw of the culturable organisms in the nanopore data. But of course, the nanopore data gave us additional insight into organisms that are hard to culture and things that we missed in our culture data. But they represented human-associated environmental inhabitants that you would make sense to be in the environment. But overall, what we took from this was NASA felt comfortable that we could use nanopore sequencing to do our microbial risk assessments because you're getting comparable results to what you're seeing with the culture data. You're not missing culture data. But this was a very limited data set. We only had a few samples, so we wanted to do some more digging into nanopore sequencing and how that looks with the microbiome. So we were fortunate to have crew members collect additional swabs from the USOS modules, areas like the air grids, the tables, the walls, some equipment. We got the swabs in triplicate and they returned them to the ground where we could run our nanopore sequencing workflow. So here's just some high-level data from that. The immediate thing that was exciting to me was we were uncovering a new core microbiome of the ISS. Across multiple locations, there were these organisms that had higher abundance. So of course, we're still seeing a high amount of Staphylococcus because that's the most abundant thing on the ISS. But we're also seeing Streptococcus, Haemophilus, Anaerococcus, Cutibacterium, and Lactobacillus, which are all hard to culture microorganisms that we're missing in our culture data. For fungus, we saw the Aspergillus and Saccharomyces that we routinely culture, but we also saw the Smollicesia species, which is a skin-associated fungal organism. So it's cool to see that. Figure 2 is just showing the top 10 suggested species, just again highlighting that we're getting much more information from this nanopore data. Figure 3 is really exciting to me because it's a Brer-Curtis dissimilarity, and we're showing that the darker colors represent areas on the ISS that are more dissimilar to each other. So it could be suggesting some ecological niches that are occurring in different locations on the ISS that could be driving diversity. And this is cool because when we look at our culture data, we don't see any difference in diversity because everything is so similar in what we're culturing. So we wanted to dig into this more and see what could be driving some of these ecological niches. So the first place we looked into is the waste hygiene compartment, which is the toilet. And of course, we saw a lot of fecal-associated microorganisms. We saw the ones that we cultured traditionally, like Klebsiella and Pterococcus, but there was a whole list of other fecal-associated microorganisms that are really hard to culture that we've been missing. So we're really highlighting how our current methods are underrepresenting fecal contaminants in the environment. Let's see. Another area that was really interesting was some exercise equipment. We actually saw some high salt-loving, borderline extremophilic microorganisms there, which is something, again, we would never see in our culture data and never really thought that they were there. But it makes sense when we start thinking about it that the crew work out multiple times a day. There could be some salt pockets building up on these equipment, selecting for that growth. Another area of interest was the crew quarters. We were seeing some high CO2-loving microorganisms in more abundance there, which these crew quarters are basically little closets that they're sleeping in, and there could be CO2 building up in little pockets there. So overall, we saw that the knowledge that we have of the ISS with our culture data is very limited, and that we can use nanopore sequencing to push forward our understanding and unlock more information about the ISS microbiome. And that we can use this swab to sequencing method to apply to future space missions, especially like when we're planning for this Artemis Gateway program. And while we still have the ISS, we want to continue to push forward the research capabilities. So we're planning to do wastewater and potable water monitoring this year. We're launching the MinION Mark 1C to the ISS this year. On the ground, we're developing air monitoring methods, shotgun metagenomics, full genome sequencing, epigenetics, and we're actually starting to get a little bit into crew diagnostics, doing some viral detection in saliva with our immunology collaborators. So before I go, I wanted to hype everyone up that we're going back to the moon, and wherever we go, our microbes go, and so will our MinIONs. Yes, so just thank you.